Hello, and welcome to today's episode on genetics and voice production in childhood and adolescence, which is a webinar held on the 30th April 2021. Adolescence is a challenging time when voices change normally and pathologically. Knowledge of voice changes related to genetic stimulation should be updated. What is new is that measurements of acoustics of voice production has increasing possibilities. Larger amount of data are necessary for measurements in variability of voice in multi-handicapped clients and many other cases. Technical methods for measuring voice productions include high-speed films combined with analysis programs like glottal analysis tools, convolutional networks analysis of high-speed films with 4,000 frames per second used in the clinical settings, as well as optical coherence tomography. Examples of these are presented in each corner. In Phoniatrics 1 from 2020, used for education in phoniatrics in the European Union, we have made an overview of voice-related genetic syndromes. Examples are given of genetic deviations for vocal fold microstructure, vocal fold bowing, vocal fold inflammation, and laryngeal carcinoma. On the next page, we will give you examples of syndromes. There are also examples of disorders in genetic neurological diseases. An example of a disorder in genetic neurological disease is spasmodic dysphonia. Two genes are given: THAP1, DYT6, 8p11.21, THAP1, C.71 plus 9C greater than A within intron 1 of THAP1, but there are many more related to spasmodic dysphonia. For all syndromes, the relation to the adolescent voice is important if the symptoms has manifested themselves at that time, and therefore deep learning is of great interest in these cases due to the big variation of voicing. There have earlier been made studies on normal voice production, and definitions of prepubertal, pubertal, and postpubertal voices have been made, but all without genetic aspects. The search for voice production and genetics development was made by the Library of the English Royal Society of Medicine. At the end, only included a few hand search papers for the last 10 years. Only a few of them were related to childhood and puberty. Therefore, we included our own intensive search of other papers, often based on reference lists and in studies of other subjects. The central activation of GnRH neurons by leptin is considered necessary for normal pubertal development. The nasal placode and development of GnRH neurons have been considered a causal mechanism as stimulating factors on key NDY neurons, kisspeptin, neurokinin B, dynorphins. The genes involved are presented in the figure. The neuropeptide alpha MSH plays a key role in energy homeostasis by mediating the action of leptin and may have a central role to the metabolic control of puberty. From the hypothalamus at the mediobasal area, the arcuate nucleus, the pituitary gland are stimulated also by the GnRH PG, gonadotropin releasing hormone prostaglandin, the reaction depending on the GnRHR receptors. Follicle stimulating and luteinizing hormones stimulate the ovaries in girls, and in boys the production of Leydig cells, production of testosterone, stimulating puberty. In girls, the anti-Muller hormone for egg reserve and inhibin level of hindering FSH production in the pituitary are regulated in balance with E2 estradiol, stimulating puberty. Extrinsic factors, for example, endocrine disrupting chemicals and body mass index, have an impact on the development. Note that voice related parameters are not included. The definition of puberty stages 1 through 5 made by Marshall and Tanner is still of interest. In a large survey of puberty by Sultan et al., voice production is not discussed at all. The author states that puberty cannot be perceived as a solitary event. They discuss the basic genetics changes. It is clear that genetic timing at the age of menarche is relevant. Sultan et al. Underline that the onset of puberty is determined by genetic heritability and neuroendocrine factors modulated by general health, nutritional adequacy, exercise, and environmental chemicals. The understanding of voice production is also related to research on transgenders. Stein has an overview without voice reference. The point being is that voice is not discussed in the literature of puberty. The table from Normal Development of Voice in Childhood, a book by Pillerson 2008, shows the puberty-related voice parameters for boys related to hormonal development divided in prepubertal, pubertal, postpubertal parts, based on age. The pubertal stages can be extrapolated. Note: serum testosterone and sexual hormone binding globulin are inversely related. Fundamental frequency, semitones in continuous speech, and the lowest biological tone are of interest. Fundamental frequency during reading of a standard text and the lowest tone, as well as voice range during reading of a standard text, are of interest in both genders. In girls, significantly related to estrogen E1 and to semitones in continuous speech and the lowest tone, among others. Change in DHEAS marks adrenarche in both genders. Maybe it is easiest in the daily work to relate to these figures of measurements related to age in adolescence. Day et al. Recognizes the biological genetic mechanisms and timing of puberty as important. In the recent large-scale genome-wide female development study, 389 statistically independent signals were found distributed across all 23 chromosome pairs. According to Hollis et al., in a male study, 76 independent genetic signals for male puberty was described. The authors found that genetically the voice break in boys was related to menarche in girls. An example of multi-handicap studies is by Schreiberg et al. They have an updated overview of percentages of neurodevelopmental disorders of speech motor speech. The advanced knowledge of today lacks perspective of voice. Interesting is a study from Sato et al. Showing that the vocal fold mucosa, unphonetic, without voice, 2 cerebral palsy children 7 and 12 years old did not have a vocal ligament, lamina propria appeared as a uniform structure, vocal fold stellate cells synthesize fewer extracellular matrix substances such as fibrous protein and glycosaminoglycan. The pubertal genetic development itself seems to be rather well understood, as shown in the first figure. The reason for difficulties for boys comparing genetics to pediatric development can be that there are several voice parameters to take into account. Technically sufficient measurements of fundamental frequency that includes registration in reading or counting. Also, measuring of the lowest tone and widening of the fundamental frequency range during reading, at best in semitones with geometric averaging of hertz. Other parameters include changes in form and structure. Thank you for watching.